Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall begin talking about cell signaling with a focus on the different types of cell signaling in this video. In the last few videos, 6.0 to 6.19, we saw how hormones such as glucagon, catecholamines, cortisol, growth hormone, as well as ghrelin, played a very important role in the regulation of metabolism during starvation. And then hormones, especially insulin, as well as leptin, played a very important role in regulating metabolism after a meal or in the postprandial state. So hormones regulate metabolism depending on the state, whether it's postprandial or early starvation. We have various hormones that are involved. We saw that the hormone, which is the signal, bound a receptor. So for example, insulin will bind the insulin receptor, glucagon will bind the glucagon receptor, and so on. And after the signal, the hormone bound or binds to its receptor, what we saw was the receptor will change shape. What we say, it becomes activated. Then we saw the activated receptor go on to affect other proteins, which in turn will affect other proteins, and that triggers what we call a cell signaling pathway. This pathway, the cell signaling pathway, will then go and affect all kinds of cellular proteins. The functions of the proteins that are present in the cell may either be increased or decreased. The amount of the proteins may either be increased or decreased by affecting transcription. Even the locations of some proteins may be affected due to this cell signaling pathway. And because of this effect on various cellular proteins, the cell will then change its metabolism. So cell signaling is a process where cells send and receive signals and respond appropriately to the signal, resulting in a change in the cellular activity. Now, cell signaling doesn't only affect or regulate metabolism. Cell signaling basically affects or regulates almost every biological process in the body. So if, for example, during human development, the fertilized egg divided to form more stem cells, and then the stem cells differentiated to form specialized cells, for example, the alpha beta cells of the pancreas, all kinds of muscle cells, all kinds of neurons, and so on. And that's how the egg gradually developed into an adult human. Cell signaling also plays a very important role in the division and differentiation of stem cells in our bone marrow. And these stem cells then go on to differentiate into red blood cells and all kinds of white blood cells. Cell signaling plays a very important role in repairing a broken blood vessel, for example, in blood clotting. Cell signaling is critical for muscle contraction. During dehydration, we should not lose any water. We should try to reabsorb as much water. Cell signaling plays a very important role in this physiological process. So cell signaling is involved, you can see, in a very, very many different processes that happen in the human body. This cell signaling allows all our 30 trillion cells in an adult to constantly communicate with one another so that the human body can function appropriately in order for us to survive. For cell signaling to occur, there must be a signal as well as a receptor. So the signal will interact with the receptor, and only then will there be a cell signaling pathway. Now the signal is mostly chemicals. So for example, all kinds of hormones are signals. The eicosanoids such as prostaglandins, leukotrienes, these are lipids. These are also signals. Of course, hormones can be all kinds, could be proteins, could be small molecules such as thyroid hormones, could be steroids such as cortisol and so on. Neurotransmitters are signals such as acetylcholine, GABA and so on. Cytokines such as interleukins, erythropoietin, these are all signals. So there are many chemical signals in our body. Then there are physical signals such as mechanical stimuli, touch, pressure, stretching, temperature is another example of a physical signal. Receptors can be divided into two types. In the first case, these receptors, what we call cell surface receptors, they are found on the surface of the cell, that means the plasma membrane. These receptors are usually what we see for signals that are water-soluble chemicals as well as the physical signals. So the insulin receptor, for example, is a cell surface receptor. The glucagon receptor, catecholamine receptor is a cell surface receptor. The thermoreceptors that respond to temperature and the mechanoreceptors that respond to mechanical stimuli, these are all examples of surface receptors. So these receptors are found on the plasma membrane. Then you also have receptors that are present inside the cell, either in the cytoplasm or nucleus or somewhere inside the cell. And these are generally receptors that respond to signals that are poorly water-soluble chemical signals. So for example, the receptors for steroids are found inside the cell. The receptors for thyroid hormones are found inside the cell, and the receptors for nitric oxide. So the focus of this video is the different types of cell signaling. We can divide this into the intracellular type of cell signaling, what we call autocrine cell signaling, and then the intercellular between the cells. And that includes the paracrine signaling, synaptic signaling, the endocrine signaling, as well as the juxtacrine signaling. Let's first look at the intracellular or autocrine signaling. In the intracellular or autocrine signaling, the signal is secreted by a cell. So here's the signal. And then the signal binds to a receptor on the same cell. So basically the cell is responding to its own secreted signal. Let's look at an example of this from the biochemistry of tuberculosis, which is a video that we'll have in the future. So here's a figure that sort of summarizes a lot of biochemical and biological steps that occurs during the infection, tuberculosis or TB. So one of the things we see in a TB infection is that these cells called dendritic cells, so we have here a dendritic cell. These cells secrete a signal called IL-12, interleukin 12. So we see the secretion of IL-12 by the dendritic cells. Now this IL-12 will then bind receptors on cells called naive CD4 T cells. So here is a naive CD4 T cells. So when this IL-12 binds the receptor on the naive CD4 T cells, these will lead to a signal transduction pathway in the CD4 T cells. This signal transduction pathway in the CD4 T cells will lead to a biological outcome. And in this case, what is the biological outcome? These cells, CD4 T cells, will differentiate into a special type of cell called helper T1 cells or Th1 cells. So basically, these naive cells will differentiate into another type of cell called Th1 cell. Now, these Th1 cells are very effective against intracellular microbes such as the TB bacteria as well as viruses. The Th1 cell now releases or secretes a signal called interleukin-2 or IL-2. And this IL-2 that is released by the Th1 cell will now bind a receptor called IL-2R present on the same Th1 cell. This binding of IL-2 to the IL-2R receptor on the Th1 cell leads to a differentiation as well as proliferation of Th1 cells into two types of cells, effector Th1 cells as well as memory Th1 cells. So this signaling that is occurring in these Th1 cells is an example of autocrine signaling. Now let's look at intercellular signaling, where these signal molecules come from another cell. We'll first look at paracrine signaling as an example of intercellular signaling. In paracrine signaling, the signal comes from a certain cell, and here's that cell releasing the signal, and the signal will now bind to a receptor on a neighboring cell. Paracrine signaling plays a very important role during the development of the embryo. Let's look at one example of this, and we'll discuss the effect of the signal nitric oxide on vasodilation. So here we have a blood vessel, and as you know, the cavity of the blood vessel is lined by cells that are called endothelial cells. These endothelial cells have an enzyme called nitric oxide synthase, which use arginine and oxygen as well as NADPH as substrate, and produces NADP, citrulline, and the gas nitric oxide. Now, nitric oxide is a hydrophobic gas. It very easily crosses the plasma membrane of the endothelial cell and then enters the neighboring smooth muscle cell by crossing its plasma membrane. The nitric oxide, once it's in the smooth muscle cell, will bind to its receptor, and the receptor here is cytoplasmic guanylyl cyclase, which is a receptor enzyme. Nitric oxide will stimulate this enzyme, and the enzyme will convert GMP into cyclic GMP. This will increase the concentration of cyclic GMP, which causes the smooth muscle to relax. And when the smooth muscles of the blood vessels relax, the blood vessels increase in diameter, leading to vasodilation. So this is an example of paracrine signaling, where a signal comes from a cell, goes to a neighboring cell, binds a receptor in the neighboring cell and causes some sort of biological outcome. Now let's look at the next type of intercellular signaling, which is called synaptic signaling. Synaptic signaling is very common in the nervous system. It's a specific and localized type of paracrine signaling. It can happen most often between two nerve cells or between a nerve cell and a muscle cell. So let's look at an example of this. We'll look at this neuromuscular junction where there is synaptic signaling between a nerve cell and a muscle cell. So here we have a nerve that is in synapse with a muscle cell. So the nerve releases the signal, and in this case the signal is acetylcholine at this synapse between the nerve and the muscle that we call the neuromuscular junction. The acetylcholine signal will bind receptors called nicotinic acetylcholine receptors, or NACHR, and these receptors are found on the plasma membrane of the muscle cell. The NAHCR receptor changes shape, and this receptor has an ion channel that is associated with it. This ion channel is going to open, and when the ion channel opens, the sodium ions, which are present at a higher concentration outside the muscle cell, they will enter the cytoplasm of the muscle cell. The entry of sodium into the muscle cell will cause depolarization of the muscle cell, and that will cause even more sodium channels to open, leading to further depolarization and eventually the formation of an action potential. The action potential that is generated at the neuromuscular junction will travel along the plasma membrane of the muscle cell and eventually reach this structure here called the T-tubule. The action potential, once it reaches the T-tubules, will cause calcium channels to open. So now calcium channels will open at the T-tubules, and calcium will start entering the muscle cell. This slight increase in calcium inside the muscle cell is going to stimulate and open nearby calcium channels located in the sarcoplasmic reticulum membrane. So the small amount of calcium is going to cause these calcium channels present in the sarcoplasmic reticulum, which is the endoplasmic reticulum of the muscle cell that we call sarcoplasmic reticulum. These calcium channels now start opening up, and that's going to release lots more of calcium into the cytoplasm. As you know, a lot of the calcium in a cell is found in the endoplasmic reticulum, and this calcium is going to come out when the calcium channels are open. These calcium channels are called ryanodine sensitive calcium channels, or RYRs, the ones that are found in the sarcoplasmic reticulum. So with this sarcoplasmic reticulum calcium channels open, the concentration of calcium in the cytoplasm will increase tremendously, and this calcium will now trigger muscle contraction. The muscle will start contracting. So this example of a signal, in this case acetylcholine, coming from a nerve, going through the synapse and binding a receptor, in this case nicotinic acetylcholine receptor, This binding causes the formation of an action potential, which then causes calcium release, and the biological outcome of this would be muscle contraction. This process is an example of synaptic signaling. Now let's look at endocrine signaling. We'll first discuss endocrine signaling involving surface receptors. As the name suggests, in endocrine signaling, the signal comes from an endocrine cell. So this signal, which is typically a hormone, will travel into the bloodstream. So the endocrine cell will secrete the signal. The signal enters the bloodstream. Here we are talking about a signal that is water soluble, so it simply travels in the bloodstream, dissolved in the water of the bloodstream. This signal will travel everywhere and it will come out of the bloodstream, and if there is no receptor present on the cell, then the signal will simply go back to the bloodstream. However, if there is a receptor present on the cell, then the signal will diffuse and bind to the receptor. In this case, the cell that has the receptor to the signal will be called the target cell. Let's look at the insulin signal and the insulin receptor as an example. In previous videos, we talked quite extensively about insulin and as a signal and how it binds the insulin receptor or the IR receptor that are present on various cells and affect metabolism after a meal. This binding will lead to the signal transduction pathway that will cause the amounts of proteins or the locations of proteins or the functions of proteins, various proteins present in the cell to be affected, and that will lead to some sort of biological outcome that we discussed in previous videos, such as increased glucose import into cells, increased glycogen synthesis, protein synthesis, lipid synthesis, as well as decreased glucose synthesis. Besides insulin, we also discussed several other signals that bind surface receptors, including glucagon, catecholamines, which are the epinephrine and norepinephrine, growth hormone, ghrelin, as well as leptin. Now let's look at endocrine signaling that binds receptors that are found inside the cells, or internal receptors. And examples of these are the steroid hormones as well as the thyroid hormones. So in this case, the endocrine cell will secrete the signal. Here the signal is not very soluble in water, is poorly soluble in water, so it will exist as a dynamic equilibrium. Some of it will be in the free form, but most of it will be bound to some sort of a protein in order to make it soluble enough to be transported into the bloodstream. Now the free form is the one that will come out of the blood vessel, and if there is a receptor present inside the cell, so here is a receptor inside the cell, the free form of the signal will diffuse into the cell and then bind the receptor inside the cell and lead to some sort of biological outcome. However, if there is no receptor present in the cell, then the free form of the signal will simply go into the cell, not have any binding, and simply come out of the cell and go back into the bloodstream. Let's look at an example of this, and we'll use cortisol. We discussed the role of cortisol in regulating metabolism during starvation. Cortisol is secreted by the endocrine gland called adrenal cortex. And this cortisol, which is a steroid, not very water soluble, so a small amount is water soluble. This is the free form. most of it is bound to a protein called corticosteroid binding globulin, or CBG. If the cell has receptors to cortisol, that would be a target cell. Then what happens is the free form of cortisol will be fused into the target cell. There it will bind the receptor, in this case the glucocorticoid receptor, and once it binds to the receptor, the signal together with the receptor will enter the nucleus, and there it will bind DNA, leading to various changes in transcription, and that causes several proteins that are involved in breakdown of proteins, proteolysis, breakdown of lipids, lipolysis, as well as synthesis of glucose to be affected, and that will lead to various metabolic changes in the target cell. Now let's look at juxtacrine signaling. Juxtacrine signaling is also called contact-dependent signaling. In this case, two adjacent cells have to actually physically contact one another in order for this signaling to occur. Juxtacrine signaling also plays a very important role in the development of an embryo. Let's look at an example of this. We'll look at Notch signaling. So in Notch signaling, so here we have one cell that has the signal, and this is called the DSL, or the delta serrate lag2 signal. So this cell has the signal, and then the other cell, the target cell, will have the receptor. In this case, the receptor is called the Notch receptor. So when these two cells come together physically, the signal will bind the Notch receptor. So the DSL signal will bind the Notch receptor. This binding is going to change the shape of the Notch receptor. It's going to become activated, and that's going to activate an enzyme, a protease, that will break the Notch receptor to form intracellular domains, Notch intracellular domains, or NICDs. So we get these NICDs formed when these two cells come in contact with one another. So these NICDs will now travel into the nucleus, and there they will alter the transcription of several genes, leading to some biological outcome, such as differentiation. Before we end this discussion on types of cell signaling, we should mention that we also have mixed cell signaling. So let's look at some examples. Let's look at autocrine or paracrine signaling, and we'll talk about this signaling pathway called Wnt pathway. This word Wnt is derived from Wg, which is wingless, and INT, which is integration genes. This was studied in the fruit fly Drosophila. So let's first look what happens if there is no Wnt signaling. So here we have a cell, we have the receptor, Frizzle receptor, or FZ, and this receptor you can see is associated with a protein called disheveled protein, shown here in orange, and there's also a co-receptor, an LRP co-receptor. Now, in the absence of the signal, of the Wnt signal, this protein called beta-catenin, shown in green, is phosphorylated, and it is destroyed by this complex protein called destruction complex, shown in purple. So this destruction complex will destroy beta-catenin, will degrade it into fragments. Okay, so we basically do not have any beta-catenin if there is no Wnt present. Now, without beta-catenin present, there is a receptor complex called TCF-LEF, TLE crouch. This receptor complex will basically bind all the genes that are affected by Wnt, and it will inhibit the transcription of these genes, and there is no biological outcome. However, in the presence of the Wnt signal, and this signal can come from the same cell, autocrine, or from a neighboring cell, paracrine, this signal will now bind to the Fc receptor together with the LRP co-receptor, and that will cause the destruction complex to be sequestered away from the catenin. Now we have this catenin that is not broken down by the protease enzyme. So this catenin concentration, beta-catenin concentration increases. This catenin now will enter the nucleus, and there it will bind the inhibitor, the TCF-LEF inhibitor. So now this inhibitor is not inhibiting the Wnt genes, so these Wnt genes get transcribed, and that will lead to production of various proteins that will lead to some sort of biological outcome such as differentiation. Let's look at another important example of mixed signaling, again autocrine, paracrine, and this is the Hedgehog pathway. This name Hedgehog or HS is named after this protein, HS protein. This protein, when it's mutated, will cause the larva of fruit flies to look like a hedgehog. That's why we have this name called Hedgehog pathway. This is a very highly conserved pathway, and again, it's very important for the development of an embryo, as well as in maintaining tissues in adults. In the HH signaling, so let's look at what if we do not have the HH signal. So in this case, when there's no HH signal, we have the receptor called the PETS receptor or PTC, and that's bound to a transmembrane protein called smoothened transmembrane protein or SMO. So when these two are bound, then what we have is we have this protein, this transcription factor called GLI. It's sequestered by this protein called SUFU. So without the GLI, without the signal, the GLI is not available as a transcription factor, and so there's no transcription and therefore no biological outcome if the signal is absent. However, when the HH signal is present, shown here by the orange ball, and again the signal can come from the same cell, autocrine, or from the neighboring cell, paracrine, this signal will bind the receptor, the PTC receptor, and this will cause the smoothened transmembrane protein or SMO to dissociate from the receptor. Now SMO will cause the release of GLI from SUFU. So now GLI is free. GLI will diffuse into the nucleus, and there it will bind to the DNA and cause the increased transcription of the various target genes, leading to some sort of biological outcome. So this is an overview of the various types of cell signaling that are important for us to understand, and a little bit about cell signaling itself. We'll have more on cell signaling in the next few videos. Until then, thank you very much for watching. Goodbye and have a wonderful day.